So everyone just be ready. I don't know what's going to happen, but
Mary Lizini from Syracuse University, and she did her PhD in the University of California, Santa Barbara. Then went from received one of those prestigious fellowships for as a postdoc in Princeton. She was then hired at Syracuse University as a professor and director of the Bioexpert Institute for very recently.
Until very recently, I thankfully stepped down.
Last year. So if I can think of someone who really is a person in software biological matter with this, for instance, among other things. That's my other things. So it's a great pleasure. Please go ahead.
Thanks for the very overly kind introduction. One of the things that I learned already is that this audience is very diverse in interests and background. And so what that means is please do feel free to interrupt with questions during the talk. Because when you're giving an interdisciplinary talk and you have a very broad audience, if you have a question, someone else has that question, and you're helping everyone else out by asking the question. So please do. And the other thing I'm going to say is, I feel like I should say now for something completely different in some sense, because I have a goal for today, and my goal is to convince you that I think that you can do physics, like real physics, at the scale of tissues. And that's a very different scale from some of the scales we've heard physics about so far. And it's not to say the other scales aren't fun too, but just I would like to sort of ask you to engage with me and ask questions to see if you believe me at the end of the talk. Okay. And so the frameworks I'm talking about today, it's kind of a long-winded title. I'm going to tell you about one example of where we've tried to understand the physics of a problem during development of an embryo and trying to understand how an organ forms during development. It's a three-dimensional epithelial structure of an organ. And so I'll talk about that later, but that's why the title is what it is. And I'll explain what these pictures mean later. You'll get it later. Okay. And so before I get started, I have to highlight the people who actually did the work. Most of the things that I'm talking about today are from a fantastic postdoc in my group, Raj Kumar Mana. Here he is. He's amazing. And he's actually going to be on the job. And Gonja is another postdoc who did some early work. Paulo Sanamantu, another postdoc. Bree Staffel, a graduate student, and Liz Lawson-Matista, a graduate student. That's mostly the story I'm telling you today. The next thing that the pointer is kind of working. So the general thing that I'm focused on right now, and I'll be telling you about later in this talk, and I'm actually not going to get to story 2. I've decided that if you're interested in plateau formation, you should come ask me about it after the talk. I'm not going to try to get to that today. But both of these problems, this is the left-right organizer in zebrafish. So obviously the inside of our bodies are not left-right symmetric. It turns out that all vertebrates have a ciliated organ that generates that left-right patterning, and I will tell you more about it, and it's completely crazy in my mind how it works, and I'll tell you about it later. But the key idea behind both of these projects is that in order to understand the function of an organ, whether it be a hair follicle, and these are simple organs, not complex organs, hair follicle, or this organ that generates left-right symmetry breaking, you really need to understand how three-dimensional morphologies emerge inside of tissues, inside of dense tissues. And you'd like to be able to understand how, for example, it changes to sort of mutations, right, generate molecular changes that then generate sort of large-scale morphological changes to organs. And that's a difficult problem, but it's one we're trying to start to address. Okay. And so the first thing I might want to point out is that if that's our goal, if we want to understand what happens to three-dimensional morphologies in tissues, then we must necessarily understand how forces are transmitted through tissues. And the analogy I talked with some of you earlier about that I really like, I'll attribute it to a biologist, Alain Mongera, who's now at University College London. I want to give him credit because he's the one who gave me this analogy, which is that imagine glass blowing an ornament. Okay, and if you have a glass blown ornament and you start with a spherical blob of glass, and then you need to end up with some elaborate morphology or some elaborate shape. What are the tools the glass blower has? Basically 2. One is some way to apply localized forces. There's a thing called a mandrel rod in English and a table that they work on so they can apply localized forces. And the second thing, of course, is the temperature. And why is the temperature key? It changes the rheology, the material properties of the glass. And very purposefully, what the glass blower does is if you need a large scale morphology change, you apply temperature right there. So you melt the glass into a fluid, you can stretch it at very low energy cost, and then it reforms and solidifies so that you can support shear stresses and be an object. And I'm going to argue today that that's very much the way embryos work. And so I'll tell you about some of the details over the course of this talk. Okay, but if you think that it's important to understand the rheology of a material, right, which is basically how do forces generate shape changes in a material, that's what the word rheology means, how forces are transmitted, just like in that glass blowing example, I would argue that many tissue types need to undergo a fluid-solid transition in order to allow those large-scale deformations that are needed for animal body plans. Plants work totally different, by the way. They're exactly the opposite of this, but for animals, this is a very common theme. Okay, so how do cells control those tissue scale properties? You know, because things, the unit is a cell. That's where all the DNA and the programming is. How do cells, a single cell, change its properties to generate this emergent fluidity of the tissue? This is a big question. And so here's a couple of beautiful papers that if you're interested in this topic, I highly recommend reading. Ali Mongera, that's the Mongera who has the glass blowing quote, and Nicoletta Petridou, who's also a fantastic young researcher. And both of these two papers that have come out maybe in the last 6 years or so basically generate that there are fluid-solid transitions. They show that what I just said is true experimentally. That basically in both of these cases, you need a flow. In one case, this is the zebrafish body axis. And so what they showed in this example is that this region, okay, so this is the tail of the zebrafish. This is the head of the zebrafish. This is tissue, so lots and lots of cells, very different scale. What they showed is up here, the thing is very solid-like. The tissue is very solid-like. Down here, it's extremely fluid-like. And they basically show that in this particular case, that it's this difference in material properties is generated by changes in extracellular spaces inside the tissue. And I'll talk about that in a moment. And this is gorgeous. A very paper by this group, it's Eduardo Mendezo and Carlos Kaiserberg. This is also zebrafish, an excellent model organism. But it's a much earlier stage. And so at the beginning of zebrafish filament, all the cells sit on top of the yolk sac. So this is the yolk sac, and here's the cells that will become the animal. And there's a characteristic time. It's almost like you can watch it in a movie. It's like a switch that all of a sudden the cells decide they have to move down the sides of the animal to form the long axis of the animal. And this is showing you that right at that time point, the system dramatically changes its adhesive properties between cells. That changes its connectivity. So these like little red things are connections between cells. And you can see you go from having lots of connections between cells to very few connections between cells. And this really is sufficient to drive a real bona fide rigidity transition in the tissue that allows this large scale flow. It's amazing, right? It's very cool. Okay. And so the next thing I want to say is actually that both of those two examples change their rigidity via kind of the same mechanism. It's a change in the effective density of the cells. So if I go from a low density, maybe you've heard of the jamming transition before. I studied that for a long time myself. And if you go from low densities to high densities, you become rigid because you make a lot more connections with your neighbor. And that extra connections really allows you to be rigid. And I'll tell you a little bit of the math about that a little later. Okay. I want to argue, and actually this is what my group is known for, is that there are some tissues where you can't change the density because it's an epithelial layer, and that means the cells fill all of space. And they need to fill all of space because they form a barrier function. Gaps between those cells would be extremely problematic to you and vertebrates in general. So the question I had when we started on this problem is if I have a cell packing like this, where there are no gaps between cells, how the heck am I going to go from a fluid to a solid, which seems to need to happen in these cell types, because there's no change in density and this thing can't happen. This kind of transition can't happen. And so what we discovered now a long time ago, like basically almost a decade ago, is that there is this feature, this mechanism, and I'll call it this has a weird word. I'll call it geometric incompatibility here. There's a few other words for it. It turns out there is a fluid solid type of transition that's allowed in this type of structure. And I'll explain it in a little bit more detail in just a moment. I think my clicker is not working. Okay, so yeah, so these examples are the first type of transition where you change the density. And it turns out that, like, if you're trying to think about a physical model for those systems, modeling them as squishy spheres works really well because the squishy spheres get the change in density right. Okay, so we need a model for this other type of transition. And the type of transition that we're going to talk about is this geometric incompatibility. So I'll just talk about it a little briefly. So what type of model? So squishy spheres are not going to be a good model for this system. So the model that was developed actually by Frank Hulaker and collaborators more than 20 years ago now is called a 2D vertex model. And the idea is as follows. If I have a layer of cells, so this is a monolayer of cells, they sort of tend to look like this in epithelia. And I can do what's like a bird's eye view looking down at the cells. And so if I look at it from a bird's eye view, they kind of basically look like maybe slightly curved polygons. Okay. And so you say, well, as a physicist, you say, well, what features of this structure can I measure? It's a packing fraction 1. But what I can measure is I can measure the area and the perimeter of such cells. And what you might think about, and there's a lot of biology that in this talk I'm going to gloss over, but I would be very happy to discuss with you. It turns out that a model like this is a remarkably good model for the mechanics of these type of tissues. So let me tell you a little bit about it. The idea is that let's say, and there's a lot of biology here. Let's say that cells via adhesion molecules like E-cadherins and via myosin and other things have a perimeter, a homeostatic perimeter that they like to regulate. I'm going to call that P0. So this is sort of chemical, basically features of the expression of biomolecules that basically a P0, a perimeter for each cell. And then I say, well, also in these cell types, the ion channels aren't so active, at least at this stage. And so the volume is basically conserved. Okay, not quite conserved, not perfectly conserved, but roughly conserved. And so if the cells are incompressible in volume and they're a monolayer, and there's things called tight junctions that basically repress fluctuations in height, then if I have a fixed volume and I have a quadratic penalty to fluctuations in height, first order, I get a quadratic penalty in fluctuations to area. So this is basically a 2D approximation to 3D volume incompressibility. Okay, and so this A naught is then regulated by volume of the cell. So it's the characteristic area that these cell types like to have. Okay, so far? Okay. So then we do the physicsy thing and we say, well, okay, there's many higher order terms here, but the simplest basically model I can write down that would have that as basically the these as the homeostatic states or a quadratic expansion around those states. So this is just some dumb modulus times a quadratic expansion of the area and some other, quite frankly, dumb modulus that is a quadratic penalty in the perimeter. Okay. And then I can tell you actually what you can measure. It turns out like you could do an expansion, of course, of this term, and it gives you like a quadratic term in the perimeter, a linear term in the perimeter. There's actually good evidence from cell biology that these terms are both there. And I can talk more about it, but I won't focus on it for that. This talk is okay. Okay, but then putting your physicist hat on again, this energy functional has basically one important dimensionless parameter. So if I non-dimensionalize this equation, basically, it's this perimeter divided by the square root of this area. This is the dimensionless parameter that sort of emerges. There's also the ratio between these two moduli times the length scale, but it turns out that's less important. And so then I can think about what this parameter is. It is the shape of the cell, basically. So if I have a large perimeter relative to my area, I'm elongated, right, in the tie length, something like this. And if I have a smaller perimeter relative to my area, I'm more rounded. Okay. And so the big surprise, so there's a ton of work on this model. Like this model, I would say this paper is if you're interested in this stuff, this paper by Hardy-Farr is a really lovely paper on the topic. Okay. So what happens? Okay, so this is our model for the system. There are reasons for it. What is the emergent mechanics of this model? Okay, the emergent mechanics of this model are that this model has a fluid solid transition. It's very cool. And I'm not going to go into all of the details for this talk because I want to get to those sort of some applications and those 3D models. But I just wanted to show you a plot, which I think is really cool. I'll tell you a little bit about the physics of it too in a moment. But I guess it's just being very slow loading. I'm sorry. So it turns out, I'll tell you a little bit about this data. So this is data from fruit fly embryos, where you can look at cell shapes and you can measure the rates of cell rearrangement. Okay, and so this axis is that shape index I told you before. So if it is low, that means I'm more rounded. I have a more rounded shape. And if it's high, I have a more elongated shape. This axis, I didn't tell you about it all because I'm a bad speaker. And I don't want to tell you about it because it's complicated, but it's basically roughly a pneumatic order parameter. And so it turns out that if I have an anisotropic system, like convergent extension in the fruit fly, where I clearly have anisotropic stresses, then I also need this in order to talk about the phase, basically the phase behavior of the system. And then, so this black line, I'll tell you where it comes from in a minute. This is a theoretical prediction with 0 fit parameters, 0 fit parameters. This is a very universal behavior. Okay, and the data on top of it is data from individual fruit fly embryos that are colored by how many rearrangements there are. In the fluid phase, you expect a high rate of rearrangements, and basically in the solid phase, you expect a low rate of rearrangements. It's basically within the noise of the system. And so the blue is the solid-like behavior in the fruit fly, and these are measurements of actual shapes of cells in the fruit fly. And this, again, I'm just going to remind you, because I don't think it will ever happen to me again. This is a theoretical prediction with no fit parameters. Okay. And it's basically like the orangey colors where it's fluid-like up here, and this is the blue. Okay. So this simple model, I think, is a remarkably, probably too good for reasons we could talk about later, model for the emergent mechanics of these types of tissues. Okay. So then, of course, if you're like me, you're like, what the heck? How did this fluid solid transition happen when I'm at packing fraction unity, right? Like why? Who ordered this? What is this universal curve? Okay. And so I wasn't gonna include this, but I know some of you here are interested in network stuff. And I did want to show a few equations to show that if you want to talk to me about it, I can tell you a lot of the details. So the idea or the question is actually, it turns out that this math I'm about to talk to you about also works in under constrained fiber networks of the kind that are also useful for talking about extracellular matrices. And they're actually, I don't know if those of you who work on network science, I've been talking to a few of my friends. I wonder if any of this might be interesting for things like social networks too, if there's some other networks in which this is interesting. But this movie was basically a movie where I changed a single parameter in one of these networks. And at the beginning, it's colored by how much tension is in the system. So at the beginning, everything is gray, which means it's floppy. And in this case, I'm actually straining the system for this network. There's a critical strain at which the system becomes rigid. But clearly the topology of this network has not changed at all. You all agree that this can't be like jamming where I change the number of contacts and that's how I became rigid. Okay, so that's the question, the theory. What causes rigidity in this type of network? And I'm going to insist that we do it in a hard problem where things are disordered, because that's where biology is. It's a little easier to do this in ordered systems. Okay, so to do this in disordered systems is what took us a really long time. I'm not going to go through any of the details of this. You don't even have to look at the equations really. I just want you to feel comforted that they're there. Because I'm basically, but you understand the problem that I'm about to do. I'm basically going to insist that we start in a mechanically stable state of a disordered system that's described by an energy landscape that has a lot of metastable minima. And then I'm going to say, well, we do the usual physics thing where we ask, okay, if I perturb my degrees of freedom, and here the degrees of freedom are the vertices of that spring network or the vertices of the vertex model, I'm going to ask, what is the perturbation to the energy? The thing that it makes perfect sense to do. And so we sort of are looking at basically the curvatures of this potential energy landscape, okay? And we perturb the system coordinates and then we write down the math. And it's basically, for those of you who have done this sort of thing before, it's just the Hessian, basically. And the condition, if something is floppy, is just that basically this variation in the energy is that this variation is 0. If that variation, if I make a perturbation to X, and it costs 0 energy, then it's floppy. That's the definition. And this is an infinitesimal definition. So we're not thinking about mechanisms or things like that yet. Okay. So this is just another way, for those of you in the know, this is just the Hessian. The Hessian describes the physics of this problem, and you can write it in this way. There's an important thing that I want to talk about for just a brief moment called the rigidity matrix, which is an important object because it discusses the derivatives of the constraint. So like a constraint is basically one of the springs between these degrees of freedom, or in the vertex model, it's a little bit more complicated of a constraint, but you can still think of it this way. And this is a very natural object. In fact, engineers that build bridges or even tensegrity structures think about it all the time. It's the derivative of the constraints with respect to the degrees of freedom. It's the obvious object to consider. And it's not square. It has M constraints and N columns, okay? So if I want to think about a first order rigidity, which is whether a first order perturbation to the constraints will cost energy. I'm going to go through this really quickly, but I'd be happy to talk to you about it later. I basically have to think of a set of displacements that preserve the constraints to first order, and therefore it's in the right null space of the rigidity matrix, if that means something to you, okay? Then if there are no first order zero modes, it's first order rigid. And if I put an extra set of constraints on my system, so you see how this thing is kind of over constrained, I have extra constraints on it. You can see that then you can basically have a set of stresses on the bonds. Okay, and if you think about it for a second, that's basically a set of forces or stresses on the bonds that don't affect the location of the constraints. That's something that's in the left null space of this rigidity matrix. Cool so far? If you're not, it's totally okay. This takes a little bit of time if you've never seen it before. But then the nice thing is all of jamming physics, the physics of squishy spheres, is first order rigidity, because if you apply the rank nullity theorem to this rigidity matrix, it basically tells you that the number of degrees of freedom minus the number of constraints have to equal the number of zero modes minus the number of states of self-stress. This is called Maxwell-Caledine constraint counting. It's how you tell if a bunch of squishy spheres are going to become rigid. That's it. It works. It works. So if you take a jar of marbles, you got to coat them in Teflon so they're frictionless, because friction is complicated. But frictionless marbles behave exactly like this. And it turns out the zebrafish examples that I gave you at the beginning that are well modeled by squishy spheres exactly follow this rule. This is first order rigidity. Okay. Then vertex models do not fit into that category. Turns out vertex models have an extensive number of first order zero modes, and they only are rigidified at second order. And so there's this sort of, and I'm not going to show all the maps, but again, if anybody is interested in this stuff, and there's a lot here that I'm sweeping under the rug. But basically, if you think about constraints to second order, it turns out that rigidity in vertex models occurs precisely because there are constraints only to second order there's second order perturbations to the constraints. Okay. And we have this paper that is horribly long, and I actually don't recommend reading it. It's bad. Well, it's good, but it's like awful to read. So I'm just going to tell you what it says, because it took us a really long time to prove this. So we proved that vertex models are second order rigid. Okay, because they're disordered, it's hard to prove. At least it was hard for us to prove, but you can prove that they are second order rigid formally. Yay, okay. And so we have this paper also has a lot of other conditions, and because this works for spring networks and lots of other networks and things too. So there's like a lot of other cases that are in that paper as well. Okay, but the key thing is that now we completely understand why those vertex models becomes rigid. It's second order rigidity, and those squishy sphere models become rigid due to first order rigidity. And so we have a complete description actually, and you can go further than that. You can talk about how the shear modulus scales with things and stuff. So basically what I'm telling you is we have a full mathematical description of both that black line that I showed you for the Drosophila data comes from thinking about this, and that's where that black line comes from. And also we can describe the rheology of these vertex model tissues. Okay, cool. Okay, that is okay, because I had to do all of that. So now I'm going to talk about the real part of my talk, but I had to tell you that we understand all of this stuff about the rheology of these tissues, because now the rheology is going to be important for what I'm going to talk to you about. So we have a very firm theoretical understanding of it, finally, after like a decade. Okay, yay. Okay. So I'm not going to talk about the mouse epithelium. I'm just going to talk about zebrafish. But both of those problems are asked the same question, which is how does the fluidity of the tissue, whether it's solid or fluid and how close it is to that transition, how does it affect the 3D structures that can emerge within inside these dense tissues? Makes sense, right? So like shape changes are going to have to be related to forces which are transmitted through the tissue and surroundings. Okay, great. Okay, and so I need to highlight all of the people who are involved. And again, a lot of this work is really, but I should also highlight all the experimental data I'm showing you is from my close collaborators, Jeff Amack, who's a developmental biologist, and Heidi Henley, who's a cell biologist. And I'll be showing you a lot of data with them today. Okay, so I need to tell you about the left-right organizer in zebrafish. This is a zebrafish at a more developed stage. You know how I told you there was that ball of cells on top of the yolk? This is still the yolk, and now this is the body. You can see it's developed a bit more. It turns out that if I look at this particular stage of the embryo, there's this organ, and my collaborators have managed to come up with mutants that label it beautifully. So there's this organ called Cooper's vesicle, and this is a sort of a face-on view of that same organ, and it lives in the tailbud of the tissue. And I'll remind you that this is the same tailbud tissue that has a mechanical gradient from solid-like to fluid-like right in this exact spot where this organ is forming. Okay. So crazy. Let me tell you something crazy. When I first heard this, I did not believe, I still am not sure I believe it, but this is the canonical explanation of how all of us end up with left-right symmetry. You have a ciliated organ. That's ciliated, so this is an epithelial layer, a single epithelial layer that forms a lumen inside of it. Sometimes in like mouse and humans, like it's maybe instead of a full sphere, it's like half a sphere. There is some differences to the geometry, but this is the problem where you have really good optical viewing. Zebrafish are transparent, so it's a really nice model system for asking. Okay, what happens is this lumen inflates, and at the same time it inflates, all of these cells, which are monociliated, they all orient their cilia in one direction. They pack a lot more cells onto the top of this frame, okay? And they pack it by narrowing these cells and stretching out these cells on the bottom. And this sets up a asymmetric flow, which is chiral, okay? And then downstream of that, and there, just before I tell you, this happens in chick, mouse, and humans, as far as we can tell. Same basic physics. Okay. And okay, yes.
Does that mean there, because for the cilia to orient in one direction with the flow, right? Do the cell on the ring has to have like a left-right series?
So what's happening is you need some, that's a great question. You basically, what happens actually is before the flow sets up, the cilia tilt a little bit. Cilia are chiral themselves. And so what's happening is the chirality of the cilia are coupled basically to a planar cell polarity. There's like a planar cell polarity pathway that allows you to basically orient all the cilia in the same direction. And that's what is the chiral feature in this system. So if you think about it, it's a way of amplifying basically the chirality of a molecule, which is cilia, basically the molecules that make up cilia, then to the level of a cell, and then to the scale of this full organ. It's kind of a really cool amplification system, actually, amplification of chirality, which is why I guess it does it that way. And the other thing I need to tell you is that, okay, how is this fluid flow read out? It's read out, there's a bunch of cells here which measure shear stresses specifically, and it knows that the flow is this way, counterclockwise, instead of clockwise, and it reads it out over here, and there's a downstream signaling pathway that does all of the rest of your left-right path. Okay. Okay. And I think like there's a bunch of interesting questions about like, how is that so robust and why does it happen that way and things like that. But I think sort of our hypothesis, so there's been a lot of actually nice work, actually mostly also by Frank Hilliker, about how these cilia get oriented and the hydrodynamics that couples these cilia together. It's a really beautiful problem. Since he's already solved a lot of that problem, the problem that I'm telling you about today is we're also interested in these shape changes that happen that pack a lot more of the cilia on the top than the bottom. Because my collaborators have shown that that actually needs to happen in order to get the downstream signaling. And the reason we think that's true, that fact is true. If you knock out, if you basically, you can apply drugs, and also there's some mutations that will basically keep everything else the same, but change so that the cells on the top aren't as densely packed as the ones on the bottom. And the reason we think it's necessary is because the readout, the amplifier readout is right up here, and the flow is this way. So you really need a strong flow across the roof of this in order to set off the sensors over here. And so we think by packing a lot of cilia up here, you can set off the sensor better. Although, I don't know if that's true or not. That's a just so story. But I can tell you that this is necessary. This shape change is necessary for downstream signaling. Oh, and this is a picture of like you can inject beads and watch the fluid flow and demonstrate that this counterclockwise fluid flow needs to happen if you're going to get correct. And if you reverse the flow, there are animals that have the flow reversed. They end up with the opposite inverted symmetry to their bodies.
Doesn't it collect motion too, combining with this effect?
Does what?
Does there is a collect motion combined with this effect in this case?
This is just the fluid flow. These are just beads in the fluid flow, and it's just a sensor down here that's reading out that flow. That's what the canonical thing is. Okay. So then, of course, the obvious follow-up question is, oh, and I'm just showing you that's where the cells are that respond to shear stresses. And so for us, in order, like, what is our experimental observable? The thing that we're really interested in that appears to be important is the number of cilia on the anterior side of this organ compared to the number of cilia on the bottom of this organ. So I'm going to be showing you that number quite a bit for the rest of this talk. Okay, and so what causes the KV cells to change their shape? This has been a question in the field for probably about at least 15 years. And there's been multiple hypotheses. So our group actually hypotheses, well, maybe there's some upstream morphogen gradient. That would be the easiest thing, right? If there was a morphogen gradient, if there was a signaling center that was on the top of this organ that generated a morphogen, and then basically the cells responded their shapes to that morphogen. And we showed with modeling that if such a thing existed, yes, it would do the right thing. But we've been searching for it, and others have been searching for it for 20 years. And as far as we can tell, there's absolutely no morphogen that impacts this. And we've checked a lot of links, sonic hedgehog, a lot of the canonical ones that you might think would be there. So as far as we can tell, that doesn't work. Okay, so then the next question is, okay, that doesn't seem to work. I just told you that this beautiful paper said that there was a static mechanical gradient in the cella, that these cells up here were a lot stiffer, well, the tissue really, the tissue up here was a lot stiffer than the tissue down here. And so we investigated whether a static gradient in the mechanical properties would be sufficient to drive the shape change using these vertex models, which are pretty well vetted. And the answer is it sort of does a little, like the static gradient does a little, but it really does not do enough. It really can't change the number of cilia on the top versus the number of cilia on the bottom. And also their group has also, they've been able to mess with that mechanics, and that doesn't seem to be sufficient to basically do this either. So we've, as far as we can tell, the static gradient in mechanics is not enough. Okay, so we don't have a morphogen gradient. We don't have a static mechanical gradient. And then, you look at movies of this thing. Let me show you what it does. While this process is going on, this organ is moving through the tailbud tissue. It's moving relative to it. It's not just the whole tailbud is moving down, but this organ is actually moving relative to the surrounding tissue. Okay, and as soon as we realized that this is what was happening, we're like, oh, if I were to take this movie and instead make the organ the reference frame and look at the tissue flow around it, physicists, what does that make you think of? This is a participatory. What do you think? Stokes flow, right? Low Reynolds. I mean, it is low Reynolds. This is exceedingly slow, right? Exceedingly slow motion, but nonetheless motion. And you can actually look at the streamlines. You can quantify them. This was a huge project to quantify the flow of the tissue around the organ. It really looks very much like Stokes flow, as you would expect. Okay, and of course, as soon as you see that, you're like, well, if this is a deformable organ, right? If it's a little soft, and I have this flow around it, I'm gonna smush the front of it, and I'm gonna extend, there'll be drag forces over the back of it, which of course, if they were there and they were big enough, would generate exactly the shape changes you see in the organ. So of course, we're like, ooh, let's see if this works. Okay, there's a picture of Stokes Slow from the famous album of fluid motion for those. Do you know this book? It's beautiful. Anyway, okay. So we need to understand whether these drag forces, they exist. Clearly there are drag forces. Now the important question is, are they big enough to cause changes to cell shape? And the reason that I think the whole field have ignored this historically, like we've all ignored it historically, is because the motion is very slow. This organ is moving through the tissue at 1 micron per minute. Okay, so, you know, drag forces are proportional to how fast you're going. So you might be like, well, that's not going to be, even if I put in the right numbers, that's not going to be big enough to generate drag. But of course, right, the viscosity enters that equation. So if the viscosity is enormous, then of course you could generate large forces with small. And I just told you the tissue is right on the edge of a glass transition. So you would expect the viscosity to diverge there. Okay, so maybe it works. We don't know if it works, but it's what I guess the first point I want to make to you about this part of the talk is that if you have a nearby fluid solid transition, then if you are hoping that you can do some force balance equilibrium models for tissues, you should probably rethink that immediately, because even very slow motions could generate drag forces that could be important. Okay, so that's the idea. Okay, so now we need a mechanical model for cell shapes and tissue fluidity in 3 dimensions. And the way we're going to do it is use our old friend, the vertex model. We're going to do it in 3D now. So we have a target volume and a target surface area, but you're familiar, these are just quadratic penalties. And we've done, a long time ago, actually, we looked at the behavior of this thing in 3D. And sure enough, you get, so this is a plot of the shear modulus versus cell shape. And it's the exact same story. If I have more elongated cell shapes, there's a critical value at which it transitions. And in 3D, it's a little bit more smoothed out. The transition point is a little bit more smoothed out by the disorder. I'd be happy to talk to you about why. Yeah.
So in those models, number of vertices in for each cell object, is it fixed or is it changing through time?
Okay, so I was gonna actually, can you hold your question for 2 slides? I was gonna tell you something which will answer your question. And I'll tell you in a moment. These sets of results are for a Voronoi model, where the degrees of freedom are actually the cell centers, and you construct the cell shape using a Wigner-Seitz construction or a Voronoi construction from the cell centers. But you could also choose the degrees of freedom to be the vertices, and I'll talk about that in just a moment. Okay, the second ingredient that we need is now we have multiple tissue types. So just like we heard from the talk from Emmanuel this morning, if I have multiple tissue types, I can actually segregate them by having differences in their properties. And so it turns out that basically for lots of good experimental and theoretical reasons, the easiest way to have 2 different tissue types is to basically put a heterotypic interfacial tension between them, which is what you said was the key ingredient this morning also in simulations also. So it's pretty standard. So you just got to put that here. And this was the work of the fantastic Preeti Sahu of my group, who wrote a couple papers on that and how that works. This is a 3D simulation. And then this is answering your question, Gil, because we thought we could get away. Okay, computationally in 3D, these models become more complex. Voronoi models are much easier to compute on a computer because they only have 1 degree of freedom per cell. But it turns out that Elizabeth, one of the former graduate students in my group, using open source code. So if you're interested in doing this, Tao Zhang and Jen Schwartz have open source code available for 3D vertex models, if you're interested in playing around. So we use their code to basically, and Elizabeth showed that although the rigidity transitions are actually the same in both of these models for somewhat deep reasons, the interfaces are not the same in the 2. So if you have 2 tissue types, and maybe it's not so surprising. It's actually kind of surprising that vertex and Voronoi models have the same rigidity transition. They do. But vertex and Voronoi models do not have the same structure as cells and boundaries. And clearly vertex models are more appropriate because they have the correct degrees of freedom for the shapes of the cells, whereas Voronoi models are kind of a made up thing that we used for computational ease. So I have to use a 3D vertex model from now on, and that makes the simulations more difficult. But luckily Raj is awesome and created a 3D vertex model with the Cooper's vesicle and the lumen inside of it. And you can simulate this with a whole 3D tissue and, you know, using our computer cluster basically at scale. Okay, so then here's the moment of truth. Like, if you drag this organ through the tissue using some realistic parameters, does it do the right thing? And the answer is, of course, I'm telling you about it. So yes, it does. It does exactly the right thing. But there's one parameter that we don't know how to match to experiments, which is basically the natural time unit. Like, we're not sure how fast this is. This is how fast we have to make it go in the simulation to get this effect. But I can't tell you what that is in real numbers that you would see in an experiment, because it's really hard to measure the biophysical quantities that are necessary at this tissue scale. So that's kind of crummy. What can we do? Okay, but maybe pause and admire the fact that if you simulate this object and you drag it through a tissue, it does exactly the right thing. Yes?
What is the right thing?
The difficult thing to measure.
So the simulation is producing the
Yeah.
program with the drug. So what is the thing that is? Was it the reorganization of the cell? Or?
Yeah, the reorganization of the cells. So basically, if I was just going to show you, if I quantify the number of cilia on it, because they're monociliated, so the cell shapes are directly proportional to the number of cilia. So if I quantify the number of cilia on the anterior and the ratio with the posterior, in wild type animals, it's about 2. And that's what we get if we drive this organ with this velocity in our simulations through the tissue. Okay, but I don't know I can't tell you the number of that velocity. That's the challenge. And as you might imagine, although tools are getting much better, making quantitative mechanical measurements inside dense tissues in embryos is still very challenging. There's a few things I'd be happy to talk to people about, but it's hard to get numbers. Okay, so what can we do? Okay, yeah, oh, this is just saying the same. You can do phase diagrams, you can do all the things physicists like. There's a region where you get a nice there's a region of phase space, so you get basically physiological numbers for the ratio. But the motion of KV is very slow. And are the drag forces large enough? How can we test it? Okay, so in the last 5 minutes of the talk, I'm going to tell you how we tried to do this. So the question we wanted to ask is, can we perturb the velocity of Kupfer's vesicle without changing anything else? Because that would be a really, like, if we could just basically slow down the vesicle, if it's drag forces, that should slow down the, basically alter the shape changes, right? And so Heidi has this really awesome, Heidi Henley has this really awesome laser ablation setup in her lab. She's like, I know, I'm going to go in and basically ablate the notochord here, because we think a lot of the motion of this Kupfer's vesicle is being driven by convergent extension that's pushing this downward. And it's just like, if I just ablate all of that, maybe I can slow it down. So we, why not? Okay, so here's an image of a typical ablation, in an experiment, because we can label the notochord and we can be sure we ablate it. But also the other thing I'll tell you is that we're very careful not to ablate the cells that are right around Kupfer's vesicle to make sure we're not changing the shapes because of some localized effect there. And when you do that, you can watch this is just an example of one of those movies where then we can still track the motion of Kupfer's vesicle through the thing. And you actually, you slow it down by basically a factor of 2 when you do this ablation. Although if you notice, there's actually a little effect when you ablate it, actually the cells they kind of like go a little faster for a while right when you ablate. And that's actually no matter where we do the ablation, that's true. So we think the ablation actually kicks off some contractility in the cells. That's why these 2 points are different. But the key fact is by the end of the experiment, you're a factor of 2 slower, okay, when you do this ablation. So, now a challenge is we wanted to measure cell shapes, because that's the thing we were really interested in. But doing these ablation experiments, you have to have a wide enough zoom lens in order to basically do the ablation of the notochord. And so we don't have really tight resolution on the cells, and it's hard to measure cell shapes with the resolution of the microscope that you need to do the ablation. So what we're going to do instead is we are going to use the shape of the lumen as readout, because it turns out that, of course, if there's drag, this thing is also flexible. And so if there's a change in the forces on this object that's being generated by drag forces, it also shows up in the shape of this lumen, which is much easier to read out. So we do a very careful analysis of the lumen at the maximum cross section of it as a function of time. Great. And so this is I just need to tell you, this is a very, very, very hard set of experiments to justify all the time my crew spent. Okay, so, and this is only half the data. I'm sure we have like 12 ablated embryos now and like 12 wild type embryos where you do these like long movies and you have to get all the fish to lay the eggs at the right time and stuff. So this is an incredibly hard experiment. And in order to measure the shape, I'll just report the radius of gyration. So if you have a shape, right, if it's symmetric, it'll have, and you look at basically the, moment of inertia, if you want to call it, of the shape. This will be 1, this shape is less than 1, this shape is greater than 1. So it's kind of a nice way to look, because you expect the thing to get flattened if it's getting smushed, right? If it's getting dragged through, it kind of gets flattened. Okay, and the results are, this is like, again, I had to do so many experiments to get enough data. But basically, like this says that for the ablation experiments, you get basically something which is close to unity, right? You get a shape that's basically round for these cell types, and you get a shape that's smooshed for the ones where it's not ablated, right? And it's at this point, pretty good. I mean, it doesn't look beautiful, but like this is a p-value. So it's actually for biology data, I think pretty good that really this ablation experiment changes the shape of the organ. Okay. And for us, that's really nice because basically this perturbation, and we've done some control experiments too. This perturbation basically, it's only changing the velocity of this organ through the tissue because it's a mechanical perturbation. It's not a biochemical perturbation. And so for us, this is really exciting because it suggests that the dynamic forces are large enough to change the organ shape. Okay, we slow it down, we change the organ shape in the way that we expect. And just to be clear, you can really be quantitative because the other thing I'll say, and then I'll be out of time, is that it's not just the notochord pushing from up here. It turns out that these cells are actually migratory here. They're also migrating towards the bottom. So there's sort of like forces coming from the top that are pushing and forces from the bottom that are pulling. You can see it. I'll just play this movie. And if for those of you who look at cells, you'll see the little lamellipodia in this movie. Like, can you see the like little wiggly bottoms of those cells? So my biology collaborators, it's a real sign that they're migratory in this direction as well. And so we can model that. And when we put it all together, if we put all the models together, we can simulate this. And we can get really the shape changes in the experiment and the simulations to be really similar for those ablations of the notochord. And then we can ablate the bottom cells and see what happens when we get rid of the cells on the bottom moving. And then it changes the shape in the opposite way, which is exactly what you'd expect from simulations as well. In other words, if we basically put the forces on the top and the bottom, and then we ablate them, and we change the forces that are being applied to this organ dynamically, then we get the changes in the shape of this organ that we predict from just a mechanical model, which I think is really cool because I think it's the first evidence that I've seen that's quantitative that dynamic forces of drag on the scale of 1 micron a minute are important for basically organ formation, shape of organs in the tissue. Okay, so I think I'm at the end of my time. I have some more stuff. I'll just stop there and I'll take questions. So maybe I'll say my conclusions just like, okay, I didn't write it down. One conclusion is that maybe if you're interested, I think there's a lot of fun physics to be done at the scale of tissues. And I would like to convince you that it's actually possible to do real physics at the scale of tissues, and that historically, like, I think people that sort of are quantitative have been hesitant to move away from things at the scale of molecules or maybe like biomolecular condensates, okay? But this is something which you can do, and you can do quantitatively. There are fluidity, changes to tissue fluidity that occur in embryos, and they're incredibly important, and they're well controlled in order to generate specific morphological changes. And I think that tissues can harness those fluidity to generate drag forces that they use to change their morphology in ways that are useful for their function. Okay. Yeah. Thank you.
Thank you very much. In order for the shape of the organ to remain there,
yeah,
you need to keep
yes
flowing through.
Yeah.
Right.
Mm-hmm.
This comes from the active force that this organ itself is making around, or?
I think it's that the nodal cord, convergent extension on the nodal cord is actually providing forces from the top. And the cells in the bottom of this organ are actually slightly migratory and they're pooling, they're actively, and actually, I don't think I have a picture, but there's some actin cables actually that I think are directing some of the motion that they're using to navigate to basically migrate towards the posterior. So it's not that the organ itself is generating the forces, there's forces being generated by the environment. And then our ablation is actually basically getting rid of those forces, and then we can see what the resulting change in the organ is.
So I imagine that if you put the organ in an environment with same viscosity, of course, you would not get that change.
Yeah.
You do not have these.
Yeah, yeah, and one of the things that is cool is there is a well, it's not optogenetic, it's an optically uncaged rho kinase that you can we have some in the lab, and so you shine a light in an area and you can actually change the myosin activity locally in the tail bud. So we think we can change and we actually have some movies, but they're very preliminary. If you change the viscosity, again, this is super preliminary, but you shine a light and you change the viscosity. We actually don't know which way it goes because we could envision it going up or down and we can't measure it. But like the KV rotates and like moves off to the side, so it's very clear that if you change the mechanical properties of the tail bud around it, like imagine that if you put like a patch of like low viscosity fluid in honey and then we're dragging something through it, it would change both the path of the object and the shape of it. And that's what seems to happen. So yeah, if you change the viscosity, you change the behavior. And if you change the active forces and keep it in the same viscosity, you would not expect a shape change, via this mechanism at all.
Very quickly,
Yeah.
you mentioned that plants behave completely different.
Yes.
Can you give a little bit?
Yeah, so plants basically don't have changes to their network topology because they have cellulose for their cell walls. So you know how I said, oh, you have to accommodate strain in order to generate shapes. But imagine that you were an elastic material and you could not have any cellular rearrangements. That's the place where plants find themselves. And so the way they generate morphology changes is actually they grow. So it's like buckling and growth and adding new cells, so cell division. I mean, animals take advantage of that as well, but for whatever reason, it seems very common across like all the bowel glands of animals that they have these very large strains where there's very little cell division. And so you really need a fluidity transition to facilitate that, I think.
I have a question. In the vertex model, how we can take into account the viscosity? Because I think the vertex model, we have vertex connected.
Yeah.
Sword.
So you can measure a viscosity in vertex models. Actually, we have a paper in preparation where you can basically oscillate a vertex model and look at the storage and the loss moduli of it and measure actually a G prime and a G double prime. So it has a true viscoelastic behavior. And I was showing you the 0 frequency limit, but you can actually, it really has a reasonable viscosity. So I'm now probing with this object a finite frequency behavior. And it really has a very, there's actually already one other paper out there by Andrei Kazmierlesh and collaborators that reports storage and loss moduli for vertex models too.
Just a remark.
Yeah.
Peculiar one. As I'm an old guy, for the vertex models, something completely different. You know that?
Yeah. Yeah.
No, we used to talk about 6 vertex models,
Oh, yeah.
8 vertex models,
Yes.
16 vertex models.
Yeah.
Exact solutions.
Yes.
Things like that. But it seems that the same data has been used in completely different.
It has. It's a problem. You're
Nothing to do.
nothing to do. And original, because the first paper came out that used the word vertex model in 2007. And some of us tried to argue that we should change the name of it for this exact reason, but it stuck. And so now we're stuck with it. You're right. You're right. It's not the vertex models from statistical physics.
Well, it seems to be the same. My soul was.
Yeah.
Change.
Borno is safer. Yes.
Safer.
Yes. You're right.
I have a very naive question.
Yeah.
Thanks for the talk. It's really impressive. Lots of energy there. Out there.
I gesture with my hands.
Sure.
Yeah.
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 Do you
foresee any type of, let's say, application of this, there's a technology there, right? To, let's say, study tissues from, cancer cells or cancer
Okay.
tissues, something like that.
Yes.
To measure the properties from tissues that are, it seems like, okay, listen.
Yeah, so we have a paper with, if you know Joseph Case, who is done a lot probably with cell rheology, where and if so, the short answer is yes, very much so. And so one of the first things we tried to do is basically ask, does tissue fluidity basically act as a marker of metastatic potential, and for example, breast cancer cell lines? And the answer is absolutely. And it's kind of interesting because a lot of the phenomena that you might think are important for cancer is like, especially for cancer metastasis, it's basically I have a maybe not all cancers are like this. Let's say carcinomas. So if I have a carcinoma, I basically have a more roughly solid like tissue-ish tumor. Yeah, it has a finite viscosity, of course, it's viscoelastic, but I basically need to pinch off some cells and they need to migrate away, right? And they're better at basically forming metastases elsewhere if they actually come off in a clump rather than as a single cell. And so you can sort of ask, okay, what's the physics that would enable that type of behavior? And it's really a viscocapillary behavior. In other words, it's a trade-off between the tissue surface tension of the tissue coming off the tumor. You can imagine the surface tension and the fluidity. And so what we actually found is that the best assay for that combination was basically tissue fusion experiments. They also use this in embryonic development, but you did basically like take 2 balls of cancer cell culture cells. And you could do this with primary tumors too, although it's a little bit more complicated because there's a lot of ECM. But if you take basically cell cultures and you put the 2 balls of cells that you put in a droplet initially so they form a round spheroid, and then you put them together, there's both a finite time scale that they take to fuse, or if they're too solid, like they'll kind of partially fuse, but they won't overcome the surface energy of those aggregates. So that's a long-winded way of saying if you do that for a breast cancer cell line, there's a very clear progression in the common, because the time scale is basically the ratio between the tissue surface tension and the viscosity, the fluidity of the tissue that sets the time scale for fusion. And that is really seems to be a good marker for metastasis across known breast cancer cell lines. And moreover, the theory that I told you that says, oh, that viscosity should be predicted by cell shape works in those aggregates as well. And so you could actually go in and look at cell shapes instead of having to do those aggregates. And Joseph, I think, has applied for a patent to go into histological sections and analyze them this way in order to test for that mechanical property. Sorry.
Question.
Yeah.
You mentioned in your talk the difference between first and second rigidity, for instance.
Yeah.
And I always hoped that maybe there was some sort of mapping between the random elastic network and the vortex model. And maybe what you're finding is that this isn't possible because they're actually losing rigidity
Different way.
in a fundamental different ways.
Yeah.
So there should be no mapping between a vertical.
Well, exactly. So the type of networks that at least I think many people have studied have been, because they've been applied to jamming, have started out with over-constrained and then diluted them down or changed the nearest neighbor networks in order to get them close to the isostat. There may be some hope for doing effective medium theories or coherent potential approximations on under-constrained elastic networks. In other words, if you really, because the key feature is that basically jamming, if you go from solid to fluid, you have to go from over-constrained to the edge of isostaticity. And that's what you're kind of approximating with those elastic networks that mostly have been studied. This thing happens precisely, actually, at it has to be at under-coordinated networks to start. And then you basically tune like a strain parameter or a shape parameter to get it. And I actually am not sure that, the models you've looked at so far won't do it, but I'm actually not convinced there might not be a good effective medium theory or something, but it would be a very different class of models because they would all start out under constraint. Does that help? Yeah, we should chat more, yeah.
More questions?
I think you studied mentioning a little bit about the plants and maybe also in the context of the cancer cells, but in a tissue where you would have like also cell proliferation and cell death. What are the expectations? Is it as simple as saying something like, well, fluidize the tissue or eliminate the drug, or is it more complicated than all these?
Yeah, it's a really good question. So that's been answered pretty carefully numerically in 2 dimensions. You basically, cell division gives you these stress dipoles, and it really acts, so like my background's in glass physics, okay? And so like, it's really remarkably the same physics that you could think about, like if you shear a glass, like how that fluidizes the glass. The physics of cell division is remarkably similar, except you just have a different elastic kernel that propagates out and effectively fluidizes the system. In 3D, it's less clear because you have another, like in 2D, there's kind of like, you always divide in the plane, and so that characterizing that is easy. It basically, it's very clear that what it does is it basically fluidizes the tissue up to the time scale on which the cell division occurs. There's characteristic time scale. And so you get just, I don't know if this is too much information, but if you look at basically so if you look at the mean square displacement of one of these tissues, if it's solid, like, you know, with a finite temperature, let's say, then you'll get a little bit of like, you know, ballistic behavior, but then there'll be an elastic plateau and things won't move. Okay, and so what you see in systems that have cell division basically is that elastic plateau gets cut off right at the time scale where the cell division happens. And then you behave diffusively on time scales longer than the time scale of cell division, essentially. 3D, it's a little less clear if there's anything interesting other than that going on. There might be, because like if you envision that there was some symmetry to the cell divisions or some correlation to the cell divisions, because you have a lot more options, you could get something that could be more interesting.
Any other question? Okay, let's thank Liz again. Speaking for today is Professor Antonio Tapilio. Who is the current president of